Give me a second. I'm opening. Not in my screen, not yet. Somebody, if somebody else is checking and signal is coming sooner, please let us know.
Okay, for me, it says we are live. Can someone check, please?
Yes, I see it on my cell phone.
It's not
No,
coming
wait,
up for
wait.
me. We're okay, Karsten?
No. I think so.
I think it's moving there, so we should be.
And that's the file from last week.
I'll check mine as well. So I think I got the. Yeah.
26 that.
Looks like it's going well. Please confirm what you have it.
We got the message.
That's the file from last week.
Okay.
It's not coming up for me. Please, if anyone in the audience has a live streaming in the YouTube channel, let us know. But I'm in the office with good signal, and I don't know where I'm doing something wrong, but I'm not getting it yet.
Eduardo, you have to go into the main YouTube channel, and it goes. The first one should be.
We are. Okay, yes, you're right.
Yeah,
yes, it's working. It's
okay,
working.
good.
Yep.
Well, we overcome the first of our issues today. So welcome everyone, and thanks for joining us for another exciting FIND seminar. My name is Adriana Maldonado Chaparro. I'm a professor at Universidad del Rosario in Colombia, and I will be your host for today's seminar. And I'm really happy that you are all here today. So before I announce our speaker for today, I would like to thank our previous speaker, David Queller, from Washington University for his great talk and discussion on shedding in the social microbiome, a total different system from the one we're going to hear about today. And because some of you have to leave early, I would like to introduce our next speaker for next week, Benjamin Finkel from University of Michigan. He will be talking about the challenges of senescence for wild male chimpanzees. So stay tuned for next seminar. Just a few reminders about our structure. We have a 45 to 50-minute seminar followed by a 60-minute discussion. And just as a reminder, those ones in the Zoom, please type in question mark in the chat if you want to ask a question after the talk is done. And then for those ones in the YouTube channel, just type in your question and then we'll have someone to relay the question to us. So we encourage you all to participate, especially students and early careers. So let's introduce our speaker for this week. I'm very pleased to introduce Dr. Aubrey Kelly. Aubrey got her PhD at Indiana University in 2014, and then she moved for a postdoc at Cornell University with Dr. Alex Ophir. He already gave a talk in the seminar, so if you want to watch that, you can find it in the previous season. Well, Aubrey, since 2018, psychology at Emory University. Her research, or her lab research, focuses on understanding the mechanisms underlying the evolution of social behavior using a comparative framework. Her lab uses integrative approaches and combined techniques from different disciplines, including behavioral ecology, neuroendocrinology, developmental neurobiology, molecular biology, and genetics. And with all these techniques, she and her lab aim to answer questions about how social behavior evolves, the variation of neural circuits within and across a species, the environmental influence of on social behavior, and the brain and social traits, variation in social traits within individual and species. So Aubrey has an impressive record of securing funding for her research, and currently she holds an NSF grant to fund her project of facilitation of group cohesion. So we look forward to hearing about those results at some point, probably some of them in this talk. She publishes actively, and her record shows a great contribution to the development of our area. You can find her work on journals such as Proceedings, Hormones and Behavior, and other more specifics to the area of research, like Brain Structure and Function, Frontiers in Psychology, Neuroscience, and Behavioral Brain Research. Well, something to highlight from her research or her work is that she's deeply committed to mentoring and training, and she has done that with around 30 mentees from undergrad to postdocs. And I could keep talking more, but I would like to introduce Aubrey. Her seminar title is Neural Mechanisms Underlying Grouping Behavior in Spiny Minds. Aubrey, thank you for accepting our invitation, and the mic is all yours.
And thank you for having me here to speak with you all. I'm going to go ahead and share my screen here. Can everybody see a slide? Excellent.
Yes.
Hopefully internet connection remains stable. I do have one of my graduate students can send me a text if I freeze, so she'll notify me and I will figure something out. If all else fails, I can probably stream things from my phone if needed, but hopefully things go smoothly. Okay, so thank you again for inviting me to speak with you all. And yeah, I'll be talking about neural mechanisms underlying grouping behavior. So there we go. Now we're live. So before we get going, I think it's kind of important to consider why animals even live in large groups. Large group living clearly isn't required for survival, given that there are several species that are solitary or live in small groups. However, there are many species that have really benefited from being able to live in a large group. And some of those adaptive benefits include having greater foraging efficiency, things like power in numbers, so reduced predation, as well as more effective homeostatic regulation, as well as things like collective traveling. So when we think about the different types of behaviors within groups, there are a lot of fun things that we can look at, ranging from cooperative behaviors like meerkats taking turns serving as sentinels to protect the group, hyenas banding together for defense against predators, group hunting in wolves. You've got really neat cooperative movements in a lot of insects like ants forming bridges so that the colony can cross greater distances. And then you also have some behaviors that for some species, ourselves included, that are really important, like shared parenting and something as seemingly simple as a platonic friendship, or just the ability to be able to get along with a stranger. These are things that we certainly take advantage of, but there are a lot of species that don't get along with strangers all that well. So most studies examining grouping behavior use birds, fish, and insects, likely because there are a lot of species in these taxa that live in large groups. We can study mammalian grouping behavior, certainly in people and non-human primates, but a lot of mammals that live in large group, like wildebeest and elephants and wolves, you can't exactly bring them into the lab, and the types of neuroscience studies that you can do with them are really quite limited. I used to study grouping behaviors in birds. In fact, all of these species on the photo on the left here, we've got Angolan blue waxbills and spice finches and little violet-eared waxbill. They vary in their species typical group size. And so for a number of years, I looked at how the brain kind of evolved across these closely related birds to see what changed to go from an ancestral kind of solitary phenotype to a really gregarious, highly social phenotype. But then I was curious about how these neural mechanisms may have not only just evolved within birds, but also across taxa. I was also very tempted by the amazing neuromanipulative tools that are available for mammals, rodents in particular, that just aren't as readily available for bird researchers. So as a postdoc, I worked with prairie voles, hoping that they would be a highly social rodent, much like a zebrafinch. And then I quickly learned that how people define highly social can vary quite a bit. So while prairie voles are fantastic for studying family dynamics and parental behaviors, and of course, pair bonding between mates, they don't really get along with strangers, and they just don't naturally live in these really large groups, especially with mixed relations. But it was my great fortune to stumble upon the Spiny mouse in 2018, coincidentally when I was on the job market. So quite lucky. So spiny mice, they can be found in groups of roughly 20 to 30 of mixed relation. They are precocial. So these little guys are born with eyes and ears open and fully furred. They can actually eat solid food on the first or second day of life. And so this kind of opens up this species for being really amenable to some cool developmental studies. As you can imagine, being able to walk around the day you're born, being able to actively engage with your social environment may really influence how you develop as an animal. And so there's some neat studies that you can do comparing altricial and precocial species with the spiny mice. Spiny mice are communal breeders, so everybody in the group is going to care for young pups that are wandering around. Females will indiscriminately nurse pups, whether they're related to them or not. And then we have found that the males, excuse me, the males are really quite paternal toward any pup as well, again, regardless of genetic relation. Something that's unusual about the spiny mice is that they accept unrelated newcomers into a group. Excuse me. Pause. Sorry. So these guys accept unrelated newcomers into a group, which is kind of unusual for rodents. Generally, a lot of species aren't going to have just, Bob walks down the street and they say, like, come on in and join our group. But it was in 2011 when a group in the Czech Republic showed that you can take established groups of spiny mice and introduce adult males to the group. And within roughly 1 to 2 weeks, the males go from hanging out on the periphery of the group to being accepted essentially into the huddle puddle with the group. And we have since seen the same thing with males and females of unrelated conspecifics being accepted into groups. Some fun facts about spiny mice is that they are the only mammal known to be able to regenerate entire suites of tissue. And they are also the only rodent known to exhibit a menstrual cycle. So spiny mice are primarily studied for their remarkable regenerative properties, and now there are an increasing number of researchers looking at them for menstruation. But we happen to be interested in them because of their group living structure and the high levels of sociality that are required for a group of animals to be able to accept something like a newcomer into their group. So when you're primarily interested in examining neural systems that contribute to grouping behaviors, spiny mice are a wonderful organism because they're not only quite social, but importantly, they're also tractable for lab studies. There are a number of species that would be wonderful for studying grouping behaviors, but it's key that they're small enough to be able to maintain in groups in cages in the lab. And so spiny mice really fit that bill for us. Okay, so if we think about behavior, how we behave is going to be really strongly dependent on the social context. So for the purpose of the study that I want to talk to you about first, I want to specifically distinguish between reproductive and non-reproductive contexts. So reproductive social behaviors are any social behavior that's in a context where behavior might be driven by a motivation to mate or protect offspring, whereas non-reproductive behaviors, you can think of them as being purely platonic. Sorry, my animations are a little slow today. So if you belong to a species that lives in a large group, you're simply going to have more opportunities to interact in a non-reproductive context. We as humans are constantly in non-reproductive contexts in friendships as well as professional settings. And something that's an important characteristic of being able to live in a large group as a species, at least in order for them to be successful, is that you need to be able to behave non-aggressively, at least most of the time in non-reproductive contexts. Of course, even highly social species certainly engage in aggressive acts. We as humans are highly social, but we're also capable of pretty egregious behavior. But generally on a day-to-day basis, when we go walking around town, we're not picking fights with people. So what mechanisms may have arisen over evolutionary time to help encourage and promote pro-social interactions in non-reproductive contexts so that it can help maintain a successful large group? One hypothesis is that a mechanism that promotes large group living is for novel, non-reproductive social interactions to be rewarding. This seems rather intuitive to us. Like when we think about our friendships, like, of course we find those rewarding. But for species that didn't evolve to frequently engage in non-reproductive interactions, they may not actually find these things rewarding. So to test this hypothesis, we use the small group living Mongolian gerbil, which has a social structure much like a prairie vole, if you're familiar with the voles. So they're socially monogamous. It's a male and female pair, and they live with usually 1 to 2 litters of offspring. So they're in these small family groups. And then, of course, we have our spiny mouse, which lives in the larger groups of related and unrelated individuals, where spiny mice will accept a newcomer into the group. Mongolian gerbils will not. If you were to try and force this, it would likely result in the death of the newcomer. You might be asking, are spiny mice and gerbils an ideal comparison? And you are correct to question that. So no, they're not the perfect comparison. However, genomic studies have shown that spiny mice are not really well named. They're actually much more closely related to a gerbil than they are a mouse. And so they are relatively closely related, and it's simply the best comparison that we can do. So in a perfect world, I would be able to go to Africa or the Middle East and do some field work on some of the other Acomys mouse species to see if there is a real close relative to the spiny mice that we study that is maybe more solitary or simply just less colonial. But I sadly don't live in a perfect world, and we don't have money to go do that. So for now, the best we can do is compare the gerbils with the spiny mice. So first, we wanted to get a profile of social phenotype for both spiny mice and gerbils in non-reproductive social contexts, specifically in the lab. So we did this first by using a group size preference test to be able to look at where does the animal want to spend their time. So there's a group of 10 novel same-sex conspecifics on one side of a really long cage, and then a small group of 2 novel same-sex conspecifics. And we just put our subjects in the middle and give them 10 minutes to run around and go where they please. So on our y-axis here, we have a gregariousness score. And so this is the time spent affiliating with that large group minus the time spent affiliating with the small group. And basically, the higher the bar on this graph, the more gregarious the animal and the more time they spent over with that large group. And you can see that spiny mice in the cream colored bar, they did spend significantly more time with the large group compared to the small group than the gerbils did. We also wanted to look at kind of the time spent in the middle, because maybe they don't want to be affiliating with anybody. Maybe they just want to kind of take things in from afar without an active investigation. So if we look at just the time spent in the middle of the arena, you can see that very little time was spent in the middle of the arena for males and females. Most of that time was simply transition time of when they're running back and forth to be able to check out the animals. Whereas with the gerbils, they do actually kind of plop down for quite a bit of time in the middle of the arena, and females more so than males in the gerbils, which is consistent with some of the other studies that we've done with gerbils in the lab, and that's that females just tend to be a little bit more antisocial and aggressive than the males. And also, sorry, I forgot to say, in this top graph on gregariousness, that's males and females combined. We saw no sex difference within either of the species. So, as we predicted based on their behavioral ecology, spiny mice are more gregarious in a non-reproductive context than gerbils. But behavior can look quite different when animals are actually allowed to physically interact. So we also wanted to look at behavior in a social interaction, again, in a non-reproductive context. So here we're just allowing 2 animals to interact freely, to behave as they will. And we did this with spiny mice and gerbils. So first on our y-axis, we have time spent engaging in pro-social behavior. The variables that go into this include huddling, kind of side-by-side contact where they just kind of hang out near each other, as well as investigation. And we can see that the spiny mice are significantly more pro-social than the gerbils with a same-sex novel conspecific. Although I think it's worth pointing out that the gerbils do exhibit a fair amount of pro-social behavior. So, you know, I don't know, maybe I would call them moderately social. I don't have the graph here, but we've done kind of the comparison between spiny mice, gerbils, and prairie voles, just when we were trying to figure out like what's the best species for which questions in our lab. And the voles are actually far less prosocial than either of these two species. But then when you look at aggressive behavior, you see something that's kind of characteristic to spiny mice, and that's that the overwhelming majority of the spiny mice, males and females, they don't engage in any aggression. Whereas then if you look at the gerbils, you are going to get most animals engaging in some aggression. Also, if you carry these tests out long enough, the spiny mice are just going to basically go plop down next to each other in a corner and just sleep together, whereas the gerbils will often increase in fighting, or they'll kind of choose opposite sides. So from looking at that group size preference test, as well as the social interaction, basically this shows that spiny mice are simply more gregarious, they're more prosocial, and they're less aggressive in a non-reproductive context than a gerbil. And again, this is what we would expect based on how they live in the wild, but we wanted to make sure we could also mirror this in the lab so that then we could go in and look at how the brain might promote these differences in behavior. So returning to our hypothesis that a mechanism that promotes large group living is for novel non-reproductive social interactions to be rewarding. If we want to test this, we can look at brain mechanisms that modulate social reward. So how does social and reward circuitry respond to non-reproductive social interactions? We can use immediate early genes like FOS, which serve as an indirect marker of neural activity, to examine responses to certain stimuli. So in this photo, we have what happens to be a vasopressin producing neuron labeled in green, and that FOS protein, that immediate early gene protein, is in red. So when a cell expresses FOS in its nucleus, that means that it's recently responded to something. So what we did was we took spiny mice and gerbils, and we exposed some animals to a social interaction with a novel same-sex conspecific, and other spiny mice and gerbils were exposed to a clump of paper clips that were the size of a gerbil and a spiny mouse. And so basically we wanted to control for novelty here and be able to assess between social and non-social. So for social circuitry, we examined the oxytocin cell group in a brain region called the paraventricular nucleus of the hypothalamus, or PVN. So there are studies that show that PVN oxytocin projections to, for example, the ventral tegmental area, this node in reward circuitry, that PVN oxytocin can influence reward circuitry. My past studies in birds showed that this PVN oxytocin cell group directly promotes gregariousness in zebra finches. And studies in voles also show that this cell group is involved with affiliation between pair bonded males and females. So together, this cell group is involved in a variety of social behaviors. Something that I won't get into quite as much is that it's a really dynamic cell group, and it's also involved in modulating basic physiology as well as the stress response. So it kind of depends where it's projecting to in the brain for how it's going to influence behavior. So we looked at this cell group because of previous studies showing that it's important for social behavior and for its connectivity to social reward regions. And on our y-axis, what we're looking at is basically how active this cell group is. So this is the percentage of oxytocin cells in the PVN that were expressing FOS. So how active was the PVN cell group? In both spiny mice and gerbils, we see pretty much the same effect, such that in the dark bars, these are animals that were exposed to a novel same-sex conspecific. Their PVN oxytocin cell group was significantly more active compared to their counterparts that were just exposed to the novel object. So this suggests that this cell group might just be processing social versus non-social, taking information about the environment. What am I dealing with here? Is it social? Okay, I'm going to be more responsive and maybe have different downstream targets, versus non-social. I'm going to talk to other parts of the brain instead. So while things look incredibly similar here between species, we can get a better idea about whether or not there might be differences in how activity in this region relates to behavior by relating the neural activity to what they did during this immediate early gene study. So here we're looking at the time spent engaging in prosocial behavior with that novel same-sex conspecific. on the y-axis, and then on the x-axis, we've got how active the PVN oxytocin cell group was. So in spiny mice, basically, the more active the cell group, the more pro-social they behaved. And this is both males and females. Again, there were no sex differences. Whereas in gerbils, you don't see that relationship. So this suggests that, okay, so while this cell group may be similarly active, if it's relating to behavior differently, then that means probably if we had increased spatial resolution, we'd be able to see that it's a big cell group. There are lots of cells in there, often like 200 to 300 oxytocin cells. They could be going to so many different parts of the brain that this activity in spiny mice may be more reflective of targeting downstream regions that are important for the promotion of things like prosocial behavior. Whereas with gerbils, we don't see that. It's possible that most of the activity in this brain region for a gerbil may have been going down straight to the pituitary to modulate a stress response, because presumably, since these guys don't normally go interact in non-reproductive contexts, it may have actually been stressful. So this is only correlational, but it does suggest that PVN oxytocin may be involved in promoting prosocial behavior in spiny mice, but maybe not so much in gerbils. For reward circuitry, we examined the tyrosine hydroxylase cell group of the ventral tegmental area. It's big, long words. So tyrosine hydroxylase, or TH, it's the rate-limiting enzyme for catecholamine biosynthesis. So these TH cells are presumably dopaminergic. And many of you have likely heard of the ventral tegmental area, or VTA. Dopamine is produced here. It's a crucial node in reward circuitry, and in a variety of species has been shown to be involved in modulating not just reward, but social reward. So we can take a look at what this cell group was doing in our spiny mice and gerbils when they were interacting with a novel same-sex conspecific or novel object. So on our y-axis this time, it's just how active is that VTA-TH cell group. And here we do see a difference between the species, such that in spiny mice, when animals are exposed to a novel same-sex conspecific, this cell group is much more active than when animals are exposed to a novel object. Whereas you don't see that in the gerbils, suggesting that for a gerbil, a novel object is just as rewarding as interacting with a novel same-sex conspecific. And given kind of how inactive this cell group was, that kind of suggests it's neither are very rewarding at all. Whereas in the spiny mice, you do get more of a reward response when they're engaging in this type of interaction. So this suggests that non-reproductive social contexts are rewarding for spiny mice, but not for gerbils. And again, we can relate how activity in this cell group relates to behavior. So here, again, on our y-axis, we've got the time spent engaging in prosocial behavior with that novel same-sex conspecific. And then on our x-axis, we have how active the VTA dopaminergic cell group was. Again, you see this correlation in spiny mice. The more active the cell group, the more prosocial the animal behaved. And you don't see that relationship in gerbils. Also, just a little note down here. I didn't relate neural activity to aggression, or rather I did, but there's no relationship, so I'm not showing it. So not only do PVN oxytocin and VTA-TH neurons respond more in the non-reproductive context in spiny mice, but both of these cell groups also relate to prosocial behavior in the spiny mice, but not the gerbils. So I had mentioned earlier that PVN oxytocin connects down to the VTA in lab mice. And one of the things about working with a species that's not common in labs is that you kind of have to redo everything. So you can't assume that, neuroanatomical connectivity is going to be the same across all species. All animals have evolved under unique environmental pressures. And so we needed to be able to confirm that, okay, sure, these two brain regions talk to each other in lab mice, but we didn't know if they did in spiny mice and gerbils. So my student, Brandon Fricker, he used retrobead tracers. So he takes a giant syringe and injects it into the VTA, and it's got these fluorescent beads in it. And basically those beads are going to travel kind of back stream to the cell bodies that kind of will start off your neuron and send axonal projections down. So what we can do is we can then take those brains that have had the beads injected, and we can look at the PVN to see, did any beads make it up there? And so we can label for oxytocin in the PVN, and then we can look there to see are retro beads present, which they are here in red. Then we can look at the two images merged. And then on the far right here, we've got our kind of zoomed in box with all of these arrows pointing to oxytocin producing neurons in the PVN that express these little red dots. And those are the red retrobeads. So every single one of these oxytocin neurons that has the retrobeads in them, that is a cell that specifically sends axonal projections down to the VTA, which means that not only is there anatomical connectivity, but then there's also definitely a potential for functional connectivity, that these two brain regions may work together to be able to either respond to the environment or modulate behavior. So because of this, we then wanted to relate what these two cell groups were doing to each other. So here on our y-axis, we're looking at basically the PVN oxytocin neural activity. And then on our x-axis, we've got the VTA dopaminergic activity. And in spiny mice, we see this terrifyingly tight correlation, such that what these two cell groups were doing was very much in sync, whereas you don't see that at all in the gerbils. So this suggests that PVN oxytocin may gate social reward in non-reproductive contexts via influences on VTA reward circuitry in spiny mice, but not gerbils. And so a mechanism that contributes to the promotion of large group living may be to have highly social species find non-reproductive social interactions rewarding. If you want an animal, ourselves included, to do more of something, you reinforce it, you make it rewarding. So it makes intuitive sense that such an interaction would be rewarding for something like a spiny mouse that evolved to live in a large group. If you want that group to be successful, it helps to make all sorts of different types of interactions rewarding, whereas that kind of relationship to non-reproductive interactions, it's just not the same for a gerbil. So in a non-reproductive group context, thinking about it in a group context, sure, interacting with a novel same-sex conspecific may be rewarding to a spiny mouse, but are some types of interactions more rewarding than others? So the social landscape of a group is obviously just going to be more complex than that of a dyadic interaction. So when spiny mice are given options of who they want to affiliate with, do they form preferences? So recent work in my lab, pioneered by graduate student Deb Ho, has been looking at novel group interactions. So what we're looking at here are social networks specifically of investigative behavior. And these are same-sex groups, and each group is, all animals are novel to one another. And so we just wanted to see, okay, if an animal is going to be in this totally novel, non-reproductive context, how do they behave in a group? And I think it's worth pointing out that we do not know that spiny mice form same-sex groups in the wild. Chances are they don't. But Karsten's group recently did show that at least in African striped mice, bachelor groups will form in the wild for males. So with some similarities in habitat and behavioral ecology, it's possible that at least spiny mouse males may form at least probably small same-sex groups in the wild. But we don't know that because we don't have the field data. Regardless, we wanted to kind of continue looking at behavioral interactions that were free of the confound of a reproductive context and that motivation to mate. So currently exploring how animals behave in same-sex groups. So generally, if you put together 14 novel same-sex conspecifics, whether they're males or females, you can see that they're really pretty interactive, and they will physically investigate one another. But if you kind of zoom in and take a look at specifically prosocial behavior, not investigation. So this is just huddling and resting near another animal, just kind of hanging out with them over a 3-hour period. You start to see preferences with each animal. So you can see it's a little hard because it's kind of small, but basically the thicker the line, the more strongly connected an animal is to one another. And so we definitely do have certain animals within these spiny mouse groups kind of picking like, you're going to be my buddy. And you'll find that some of them will spend most of their time together. And it's also really curious that with these social networks, we can see that there are some isolates. And so there's definitely a lot of individual variation in how these animals are behaving in a group. Again, not surprising. There's lots of individual variation in how we as humans behave in groups. So from these early social network studies, we decided to examine whether there are some basic social preferences in novel groups. So Deb conducted a different set of novel group interactions that were in all male groups, where there was a mix of novel kin and novel non-kin. So in communally breeding species, it's typical for the males to be the sex that's more likely to disperse and have to find and join a new group. And interestingly, in these all-male groups that are composed of all novel, but some kin and some non-kin, males spend more time investigating and affiliating with novel kin compared to novel non-kin. So one, they've got great kin recognition abilities, which is good. Most animals should. But it makes sense to not only be able to recognize your kin, but to also be able to adjust your behavior accordingly. If this were a mixed sex group, it would be pretty important to identify kin from non-kin, for example, for mating purposes to avoid incest. But even in a same-sex group, it could be important for things like nepotism. So graduate student Brandon Fricker, he decided that he wanted to look at how the brain was differentially processing kin from non-kin in the spiny mice. So previous studies have shown that a region called the lateral septum, which is basically a region that's emerging as a hub for all things social, that it's involved in social recognition. So we wanted to determine whether the lateral septum may be involved in kin recognition in the spiny mice. So the LS is a big region. It's pretty standard for people to divide it into portions, at the very least for dorsal and ventral. If you have really fantastic whole brain mapping in like a lab mouse, you can see that you can divide the lateral septum up even further. But we're just sticking with dorsal ventral for now. And then Brandon conducted an immediate early gene study similar to what I talked about in the first study, except instead of co-localizing that FOS protein with oxytocin and dopaminergic cells, he wanted to get just a broader picture of like all neural activity in the lateral septum. And so FOS, importantly, shows up in a lot of different cell types. It can show up in astrocytes and glia as well as neurons. And so Brandon wanted to specifically get neurons. And so you can do that by co-localizing with NUN so that it's a specific neuronal marker so that you can just see specifically neural activity in the lateral septum. So we wanted to control for novelty and familiarity. So there are several studies that are looking at kin recognition, and they often expose animals to a familiar sibling, the kin. And then because they're not highly social species, they're not just living with non-relatives. And so then they expose them to a novel non-relative as the non-kin. And then it's really hard to disentangle whether or not you've just identified kin recognition or novelty detection. So Brandon conducted two different immediate early gene studies here to try and disentangle this a bit further. So in the first one, he took spiny mice, and these are all males, by the way, and that's largely due to availability of animals in the colony. And he exposed them either to novel non-kin or to familiar non-kin. So this is basically a test of novel versus familiar. And then in the second immediate early gene study, he exposed them to novel kin or novel non-kin. And so we can control for novelty and familiarity. And this one is just, do you recognize kin from non-kin? So if we take a look at neural activity in the lateral septum, that's on our y-axis here. And then on our x-axis, we have dorsal and ventral portions of the lateral septum broken apart. And in both portions of the lateral septum, animals exposed to the familiar non-kin in light green, and in the light burgundy, shall we say, color is the novel non-kin. Basically, there's no difference. So the lateral septum is not differentially responding to novel and familiar. But if you look at that second immediate early gene study where it's kin versus non-kin, you do see a difference. So just in the dorsal lateral septum, this part of the LS is more responsive to novel kin than it is to novel non-kin. So at least part of the lateral septum is responsive to kin. It's involved somehow in kin recognition. So if you look at, okay, what are the animals doing when they interact with novel kin and non-kin, we see a behavioral difference such that males are more prosocial when they interact with the novel kin. So these are all in just that dyadic interaction, but this basically mirrors what Deb had found in the group interaction. When all the animals are novel, if you're a male spiny mouse, you pick apart who's my kin, who's not my kin. I'm going to be friendly with everybody, investigate everybody, but then I'm going to preferentially be more prosocial with the novel kin. So spiny mice, at least males, exhibit kin-biased behavior and behave more prosocially with kin than non-kin, and the dorsolateral septum may facilitate this kin recognition. I see something in the chat. I just want to make sure that I'm being heard. Okay, good. Didn't want my internet to freeze. Okay. So what Brandon has done then is inject more of these tracers, viral tracers, into the lateral septum. He's done both retrobeads, but also anterograde tracers. To see, okay, great. So let's say the lateral septum is processing information about kinship. It's got to send that information somewhere. And so what he's done is identified where does the lateral septum send information to? A lot of brain regions, but two in particular, mainly one that I want to point out is in the hypothalamus, and that's our PVN, which is what I talked about in the first portion of the talk. So one could hypothesize that the lateral septum is really important for processing conspecific type information, like is this kin or not? And then it can send information to the PVN, which then can send information to reward circuitry to reinforce context-appropriate prosocial behavior. So ongoing in future studies, our PVN, we already saw from the first study that it may gate social reward via the VTA in spiny mice. But if you think about what is the VTA doing, the VTA modulates reward by releasing dopamine into the nucleus accumbens. Again, nucleus accumbens, big node in reward circuitry. But what's interesting is that PVN, it also sends direct axonal projections to the nucleus accumbens. So even though it can mediate reward by going through the ventral tegmental area, it might be able to do that by directly talking to the nucleus accumbens. So one then can ask, are interactions with different types of conspecifics differentially rewarding? We just saw that spiny mice prefer to affiliate with novel kin in a group context. So are interactions with kin, whether they're novel or familiar, are they inherently more rewarding for a spiny mouse? So Brandon is starting to get at this question with a technique that he validated called fiber photometry. And so fiber photometry allows you to put a probe in the brain to be able to measure real-time calcium signaling, so basically neural activity, to examine, in this case, it would be reward processing in spiny mice doing a number of different things. And so what's really nice about this technique compared to like the FOSS is that with something like an immediate early gene study, you only get one snapshot in time to look at in the brain. Whereas with tech like this, you can do a lot within a single subject. And so you can take your spiny mouse and you can have them interact with novel kin, with familiar kin, with novel non-kin. You could do a reproductive or a non-reproductive context, and you can start to differentiate between all these different conspecific types. Do spiny mice find some more rewarding than others? So we predict that interacting with novel kin will simply cause a greater neural response in the nucleus accumbens compared to interacting with novel non-kin. Also, a new graduate student, Christian Escalon Rodriguez in the lab, he will use chemogenetic technology to be able to inhibit this PVN to nucleus accumbens circuit to determine direct contributions to kin-biased prosocial behavior. And so some of you are likely familiar with DREAD technology. We have Cre-dependent DREADs. So basically you can inject Cre in your downstream site, which would be the nucleus accumbens, and your dread virus up in the PVN, and it connects the two so that then you can temporarily, with a peripheral injection, turn off this circuit to see how behavior changes. And then, because it's only temporary, again, you can do kind of more within subjects designs with this. So this will allow us to be able to determine basically if PVN projections to the nucleus accumbens are important for some aspect of reinforcing prosocial behavior, maybe in a kin-biased manner. If you turn this off, then you determine whether it's necessary. But it may be that it's usually never just one circuit does not like modulate a complex behavior like prosocial behavior. So we wouldn't expect prosocial behavior to like be abolished, but simply just to decrease, showing that, yeah, it's involved in this, probably as are many things. It's kind of like job security, lots of circuits to probe. So to wrap things up, there are going to be various mechanisms that contribute toward promoting grouping behaviors. Some of these mechanisms may be more ecological in nature, such that maybe an animal lives in a large group simply because they're more likely to survive having power in numbers. But whatever the reason is, there still needs to be something in place to help reinforce and promote grouping behaviors. And one way to do this is to evolve specific adaptations to social and reward circuitry to reinforce interactions with a variety of different types of conspecifics in both reproductive and non-reproductive contexts, but maybe to still favor certain types of relationships like kin relationships that might increase fitness. So with that, none of this would be possible without my wonderful lab, as well as our collaborators and of course, funding sources. And with that, I look forward to any questions and a discussion.
Thank you, Aubrey, for such a great talk. I think we're all super excited about the discussion part. Just a quick reminder, just type in your question mark and don't forget to state your name and affiliation as we're trying to build a community. So our first question comes from Eduardo.
Thank you, Adriana. Thank you, Aubrey. Fantastic talk. I have so many questions and things that I want to discuss with you. I'm sure we'll have time later, but I wanted to start with something that hopefully will provide more context to everyone. I'm listening to you and I'm thinking of the 40-year-old history of the development of the voles as a system, right? And sometimes I tell people who doesn't know the story how it all started in the 70s and 80s with Getz doing some field work. Can you just share with us? So it's fantastic. I mean, I can picture you have decades of research going forward, all the opportunities the system is offering you. How did it start? So I was checking a little bit the literature, and it sounds like there was a lot of predator-prey studies in the 90s and 2000s, and they seem to be an interesting model for circadian biology. So what is the history of when people realized that spiny mice had this kind of interesting social system? And so what is the history of field work that laid the foundations for this?
Yeah, so Richard Porter began studying them in the late 70s, early 80s, and he was looking at a bit more of their communal breeding structure. And so he was looking at, whether the pups can identify their mom from another female that's just nursing in the group, whether the moms exhibit preferences for who they're going to nurse to, and generally, he was finding that they really are kind of indiscriminate in their parental care as well as in like who the pups choose. And so he really kind of started to establish this is a viable organism for looking at communal breeding structures. And then there were a few groups that did a little bit on predator response. And so there are 2 studies that show that spiny mice, at least in a lab setting, don't respond to predator cues. But then there's one other study that says well, maybe not. And so that kind of seems to have fallen to the wayside. And then really the spiny mice took off for studying for regenerative properties, as well as as a model for diabetes. In the lab, they have a tendency to overeat and get quite chubby when they get older. And so a lot of people use them for diabetes research. But you can't order these animals, as many people in this room know that there are many of organisms that you just can't order off a shelf. And so it was the field biologists who were going out just to collect these animals for their studies for regenerative biology or whatever, to then they're collecting them in groups. Like, oh, these guys are in groups. And there's some nice studies recently out and still yet to come out that are doing kind of more genomic analyses on the distribution of spiny mice. But it was them who really kind of stumbled upon, okay, they're super social. You've got a little bit of literature from the Porter lab, who stopped working with them at some point. And then I stumbled upon them when I was on a job interview, and I was being overly honest and lamenting about how the prairie voles weren't the social bird that I was looking for. And it was Ashley Seifert, our now collaborator, who is a regenerative biologist, who said, I think you need to come check out the mice in our lab. And the rest is history.
So I'm sorry, I didn't get the last name. You said Richard Porter.
Porter, P-O-R-T-E-R.
Thank you so much.
Thank you, Eduardo. Our next question, well, I'm going to skip Rajavendra just for the sake. We have a student next on the queue. So I'll welcome Ana Maria, and then I'll come back to you, Raj. Ana Maria, you can ask your question.
Yes, hello. My name is Ana Maria Romero. I'm currently an undergraduate at Universidad del Rosario, working with partner preference in guinea pigs. And I have two principal questions. The first one is that, as you say during the talk, the FOSS method of gene expression is like an image in time, and it is like only one point in the data for individual. So my question is, if you have studied or you have some idea of the personality traits that would affect the results.
Like how might just individual variation in an entire behavioral phenotype influence that neural response? Yeah, that's absolutely something that's going to happen. Actually, my graduate student, Deb Ho, is very much into personality research. So quick plug for her, stay tuned, that she has a project planned that will be looking at how the personality of an individual is going to interact with just how an animal behaves in a group context. But generally, when you think about a FOSS response, that, this is where we need ample sample sizes, that it's going to hopefully kind of wash out between the two groups, that you're hopefully going to get just a natural balance of different personality types in animals that are exposed to the social context versus the non-social context. So you're kind of definitely operating on this kind of even layer of noise. But if you can detect signal above that, then that suggests that there's at least something real with, okay, if the PV and oxytocin cell group just responds more in everybody to a social compared to a non-social context, that sure, this cell group may also be influenced by the personality of an animal and certainly its early life experiences, but like at some basic level, like one of its primary jobs is like process social or non-social. And so, yeah, basically the bigger sample size you can have, the better.
Aubrey, I think we lost you. So we're going to wait a bit for you.
You're muted, Adriana. She didn't hear you say that.
Okay.
We lost
You're
her.
back.
We lost
Sorry,
her
I'm
for a
back.
few seconds, Aubrey.
Yeah, you
If
first.
you want to repeat the last few seconds, we lost you for this much.
I disappeared into the ether. I think the last few sentences was largely just saying that, if there's always going to kind of be that background noise, but if you can have a big enough sample size and detect a signal above that noise, then what you're looking at is a real response to something. And then, it would be amazing to be able to, before even looking at neural responses to, okay, is the brain processing, is this social or not? To be able to behavioral phenotype an animal, to then see, okay, how does their personality influence that neural response?
Yeah, thank you. And the other question that I had is like more related to the methods using for the tracing and the connection between the parts of the brain. I'm not very familiar with it, so I have a question with it. Is that to prove the connection between the PVN and the VTA, you did like the backstream connection, but for proving the LS to the PVN, you did downstream. So I was a little confused. Why did you change the method, or is there a reason between in that?
Yeah, honestly, the retro beads work better. They go back and they'll fill an entire cell body. And so it's really obvious of like, ooh, that was the source of where this came from. In the lateral septum example, we have the anterograde tracers. That's just the one that I talked about. What Brandon actually did for that is he injected a cocktail of both the retro bead and the anterograde tracers. And he did that because he had a bunch of immediate early gene data showing that the lateral septum is really crucial, not only for promoting the kin bias, but also for promoting just that basic drive of a spiny mouse to go affiliate with a large group. And so basically, because this brain region seems to be so important in various behaviors that are important for grouping, we wanted to know basically everything that's connected to the lateral septum. What sends information to it? What does it send information to? And so it had both in there, and I just told you about the anterograde tracers.
Okay, thank you.
Thank you, Ana Maria. We're going to go with Rajavendra, and then we're going with Bakudo, right?
Okay. Can you hear me?
Yes.
Yes, I'm Raghavendra Gadakar from the Indian Institute of Science, Bangalore. Thank you. This was very rewarding because it was very novel for me. So that was very nice. I have a couple of questions, both roughly about the earlier part of your talk. You showed us the neural mechanisms that make social interactions more rewarding for some species and less for the others. I can see how this can promote social behavior. But going into further detail, I was wondering the contrast between social and solitary versus the contrast between large groups and small groups.
Mm-hmm.
Will this explain both? Of course, one can think that if you're in a large group, you have much more social interaction and fewer social and small groups and none in solitary. Is that enough, or would you think that there may be other mechanisms to make the animals prefer a large group? Because in your experiment, you had this one mouse sitting in the middle and there were 2 here and there were 4 here. So to choose 4 over 2, is this difference in the reward adequate, or do you think there was something more going on?
Right. And so it was 10 versus 2, but it's still, it's an excellent question. And I mean, it's even a question of like, do spiny mice count? Do gerbils count? And so presumably they're not presumably, they are telling a difference between 10 and 2, given that they still exhibited the preference, especially in the spiny mice, in a direction we would expect. But it's actually something that as a lab we've talked about, of kind of titrating that out to see when does it make a difference. If you're going, would 3 versus 2 be enough? 4 versus 2, kind of scaling up. And we haven't done those experiments, but it's definitely something that is on our radar of trying to figure out what that tipping point is. And again, to your point, it's probably going to be a different tipping point for something like a spiny mouse than it would be for a gerbil, which is social, but in these small groups. And then if you were to take a totally territorial solitary rodent, one may be more than enough.
Right. My second question has to do with the contrast between the proximate mechanism, which you showed us about how being in a social group is rewarding, versus of course, the ultimate evolutionary explanation as to why you live in groups, because you get more lifetime reproductive fitness if you do this. So do you have any thoughts about how one can combine these two, or how, or even better, how one can find out which came first? So it become more rewarding, so I stay there and I get more fitness, so I continue, or with 2 species may be difficult, but I'm wondering if you have 3 or 4 species which live in different size groups, do you see any prospects for trying to understand which came first, which came next, and how the feedback between the proximate and the ultimate might work? Yeah,
I wish I had an answer for that. I don't. I know basically if we had the taxonomy worked out really well and we were able to kind of trace back the ancestors of the particular species that we're looking at, we might be able to stand a chance of going from more of that ancestral state to see what's changed along the way. That's what my graduate advisor, Jim Goodson, had done with estrilded finches, is that there were a range of these closely related birds that you could trace back of like, okay, the ancestral state was to be territorial and solitary. And so then what changed in the brain over time? He actually didn't spend a whole lot of time looking at reward mechanisms. But yeah, we don't have the ability to do that yet in spiny mice. I would definitely need to recruit a collaborator who is well-versed in the field to be able to go track these little critters, and then to have somebody try and help us work out who came first.
Yeah,
it's tough. It is the chicken or the egg.
Exactly. Yeah, thank you.
Thank you, Rakavendra. Well, Facundo had to leave, but he'll meet with you later. So he'll ask the question when he gets the chance to talk to you. So we have more students down, but I will call Birgit. Hi.
Thank you. My name is Birgit Sabo. I'm a postdoc at the University of Bern in Switzerland, and I study social lizards, actually. So I have two questions more related to the behavioral ecology. First of all, are there any studies that show the kin structure in natural groups?
So studies, no. Anecdotal data, yes. But yeah, we're really lacking. It's something that I'm very envious of the vole world, of the beautiful behavioral ecology that's been worked out from decades of field studies. And we do not have that in the spiny mice. And it's something that is sorely needed, hopefully someday. But it's a mixture of kin and non-kin in some of the groups that they've tracked from.
Thank you. And my second question was, maybe it's a bit out there, but is there any evidence for same-sex sexual behavior in these mice?
I do not know. There's just a really neat paper that came out, I think maybe in Nature Communications, that was looking at same-sex behaviors across a variety of species. And we just don't know for the spiny mice.
Thank you so much. I really enjoyed your talk.
Thank you, Birgit. Okay, now I come Diva. Did I pronounce that correctly? Hopefully.
Yeah. Yeah. Hello, everyone. My name is Sia Bonga. I'm from the Suckland Karoo Research Station, South Africa. And my question is on the, especially when you're talking, the last of the presentation, when you're talking about applying inhibitors, and when you're applying them, I just want to know, when the mice actually form like groups? And if they do, is this due to the maybe spending more time together, maybe like grooming and huddling? So rather than, yeah.
If I understood your question, you're asking if they form groups, or did I hear the word inhibit somewhere? I'm sorry, I missed the first part.
So when you said in your last part of your presentation, when you said one of your students is actually applying inhibitors.
I see. Yes. Okay. Yeah.
Yes,
so in the process of inhibiting that particular circuit, Brandon has inhibited a different circuit, one that is actually a connectivity with the lateral septum, and he put them in that group size preference test. And the preliminary data suggests that it actually flips the preference, such that it made them less gregarious. Such instead of spending all the time with the large group, they then, when you inhibit this circuit, it's the anterior cingulate cortex projecting to the lateral septum. All of a sudden, they spend more time with the small group. And so it's, what that circuit is doing, we aren't necessarily certain yet. I mean, it could be doing something as simple as decreasing social interest of, I don't need to go investigate 10 animals. In this case, it was, I believe, 8 animals versus 1. So instead it's I'm still highly social. I want to be somewhat social. I don't want to be alone, but I don't need to be with that big group. So what exactly we're changing, we don't know. We're still trying to hone in on that, but you can see that when you inhibit some of these circuits, you do reduce that preference to go hang out with a group.
Okay, cool. Thank you.
Yeah, Jim.
So, yes. Yeah, hello. My name is Jim. I'm a PhD.
I can't hear. I don't know if that's just me.
I cannot hear you.
Okay. So can you hear
Now
me
it's
now?
coming. Now, yep.
Okay. Because I think it froze. Okay. So I start again. So I'm a PhD student from the station. I work on the personality of the bush crew rat. So my question is actually a follow-up of Anomalia. So in the behavior, when you test an animal in their behavior, like for personality or other behaviors, it's always common that you have a big individual differences. I wonder if you have similar like variation in terms of the brain signals. Like are you also getting strong variation when it comes to behavior part?
Yeah, so I can tell you a little bit more about some of Deb's data of looking at these novel group interactions. And so she's got male and female, all same-sex groups, and she recorded their behavior during a 3-hour interaction. And then she has all the brains from all those individuals. And so what she's done is she's got a profile of, okay, how is each individual, how pro-social are they? How much time do they spend investigating? Basically, how socially bold are they at the beginning of some of these interactions? And then she's looking in the brain at oxytocin and vasopressin neuroanatomy to just look at the densities of these cell groups. And so she's correlated their behavior in this group with their neuroanatomical profiles. And there are some brain regions, one of them is the anterior hypothalamic vasopressin cell group, how basically the more cells you have in this brain region, the more socially avoidant an animal is. And so you can start to capture how the brain reflects some of these personality differences in behavior. And, at some point, you could test this causally by going in, and this is what she hopes to do next, is to go in and manipulate, okay, if an animal has a lot of AH vasopressin and they're socially avoidant, if I knock that down so they have less of it, will they become more socially bold? And so, yeah, there are kind of different ways to be able to get at how the brain relates to these different personality types.
Okay, thank you. That's very interesting.
So we still have another question from Precious.
Hi guys, my name is Precious and I'm currently a research assistant here at SKRS. My question is more on the fiber opto, the device that you use to measure the calcium. I just wanted to find out how invasive it is, and if it is invasive, how do you bypass the ethical clearance?
Yeah, so we're under strict rules and regulations via the what's called the IACUC here, the Institutional Animal Care and Use Committee. So everything we do, we first have to receive permission to justify that we have a reason to do these things. And so we're sticking an incredibly thin little probe into the brain. And so when an animal undergoes surgery, it's got anesthesia, it has medicines, analgesics, so that it's kept comfortable. And then they recover from the surgery extremely quickly. They wake up from anesthesia within 5 minutes, and they'll actually go ahead and eat and drink. And so they recover very quickly. And they're very good at being able to live with these small implants in their brains. The nice thing about spiny mice is they don't seem to be bothered by it at all, such that they're never messing with it. With rats, I know I have colleagues that have to put like cages on their heads, because like rats, they just like reach up and just like pop it off. Like, I don't want that on my head. And so the spiny mice seem to handle these small implants really well. And they just hang out happily in their cage. And then when we go to test them, there's a little fiber optic cable that you just connect it. And then that's when you can start measuring this signal while they run around. And then you just disconnect it. It's very much just like plugging a cable into your phone to charge it. It's kind of the same idea. So it just plugs into this tiny little implant. And then they run around, you get some signal, you unplug it, and then you send them home.
Okay, thank you.
Okay, so I also have two questions. So one is mine and the other one is from Carsten. So I'll start with mine. So my name is Lindelani Makoya, and I'm a PhD student with the University of the Witwatersrand, as well as a research manager here at the Circuitry Research Station. So my question is, I'm sorry if I missed this speak at the start of your talk, but the population that you're working with, is it a complete, fully lab-based, or is it something you got from the wild that you then brought into the lab? And if it is from the wild, like where exactly is their distribution?
Yeah, so the animals that we have in our colony are descendants of Ashley Seifert's colony, where he goes out into the wild to capture them and bring them back. And so he caught these animals, I believe, in Kenya and Tanzania. But the Acomys species, they're really widespread. They're throughout Africa, the Middle East, and into Southeastern Asia. There is a population of Acomys cahirinus living on the campus of University of Tel Aviv. So they're quite widespread. But yeah, these ones are descendants of, I believe, Kenya, but it could be kind of right that border of Kenya, Tanzania.
And have they been also observed in the field to show the similar group living behaviors like you see in the lab?
Yeah, so we don't have like nice experimental field studies. And so everything we know about them from the field are it's anecdotal evidence from the researchers who go out there to trap them. But hopefully someday we'll be able to get some field work. I just need to convince funding agencies.
Yeah, that's always the challenge. And then the question from Carsten is, you mentioned that they are able to recognize their kin. So he wanted to know by which cue do they recognize the kin? For example, in the mice, they always use the major urinary protein for the house mice. So is it something similar also in the spiny mouse or something completely different?
Yeah, we don't know, but presumably, there's some sort of pheromone that they've got to be picking up on. This paper actually just came out yesterday in Scientific Reports. And what Brandon found is that when they're investigating the novel kin versus the novel non-kin, they spend more time at different locations of the body. And so what we had done was we took some tissue samples and gave it to a pathology core to be able to look at where are scent glands. Sadly, they're not the same on every rodent. In gerbils, there's this nice big scent gland like right on their belly that you can see. And if you just look at the spiny mice, you can't see anything. And so we've sent tissue off to a pathology core. And sadly, the results were inconclusive. So they clearly have them, but we just don't have the data yet to know exactly where the information is coming from that may be giving the most important cue of, yeah, I'm kin. I would think that, MUPs have to play a role in this, but we just don't know that for certain.
Okay, good. Thank you again for the wonderful talk.
Thank you all for participating in the lab. That's great. And we encourage other students in early career to also ask questions. Don't be afraid. Our next question comes from Eduardo.
Thank you, but definitely skip me. But I don't see any other students coming up. I have lots of questions. This is fascinating.
Just me.
Yes, I mean it's like traveling back in time with things that were happening 20 years ago with the voles and all that. So I have lots and lots of questions, and I just want to preamble a little bit. It's fantastic. My understanding is that you're just developing this, and the sky's the limit. Because we're really just going through all the steps that people had gone in the past with the voles, and now with the titty monkeys, et cetera, et cetera. So it's fantastic. And so I don't think you have to let the audience know that there is no perfect comparison. You'll struggle with all these stupid things that people will tell you reviewing your grants. that you cannot compare gerbils with mice, and they are using mice for developing a human vaccine. I mean, I get that all the time. So it is what it is. You're making progress, and in 20, 30 years, it'll be the time to see how much progress you've made. So in that sense, laying out a very simple contrast is the way to go, which is what I think you're doing. But along those lines, I wanted to ask you, even when I'm acknowledging that you have to start with very simple contrast that will generate new rounds of more specific experiments. The first part of the talk, you were talking a lot about non-reproductive context and bringing in the human component for us to connect to those social interactions that are non-reproductive. And that's a theme through your talk. Everything is binary. It's king, non-king. It's social, no social. It's reproductive, not reproductive. But we know that none of those are binary dichotomous. So do we really have, even among humans, I mean, the non-social interactions that people have in a bar, are they really non-reproductive, or are just the early stages of a courtship process where you're assessing potential mates? So what do you mean by non-reproductive for the experiments you did?
Yeah, and so we're specifically looking at, when you think about human sexual behavior is wildly complex, there's a big push culturally, at least here in the United States, that things aren't non-binary, that, gender is fluid and sexual preferences are fluid. We don't know if that's the case for something like a mouse. If you look at, behavior of most non-human animals, you primarily see heterosexual relationships. And so that's kind of what we're referring to as like non-reproductive, is like when there is no possible motivation at any stage to mate. And so if you've just got males interacting with males, okay, you've at least removed the confound of what you're saying in a bar. Maybe it's the early stages of courtship. And so presumably in something like a spiny mouse, we're not seeing early stages of courtship. If anything, maybe we'd be seeing early stages of something akin to friendship. And I know that some people have a problem saying that a mouse could have a friend, but they might have something friend-like, they certainly form bonds with other individuals. So when I refer to a non-reproductive context, it's very much going to be same-sex interactions.
My question was not coming. I'm glad I asked, because I missed. I mean, you must have said that these were just males and males. And my question was not coming from what I perceive as a non-scientific approach no. I mean, I do believe in the biology of sex. I do believe that in terms of evolutionary biology, we have males and females. And I'm not discussing the cultural components of spiny mice interactions. No, I just missed that you had told us it was males and males.
Yes, okay. All of these, all of them were same-sex interactions.
Right. I'll see if, I have more questions, but I want to give somebody else the chance. If I know that Adriana wanted to ask you something, so thank you.
Thank you, Dardo. I'll give you the mic after my question. I have 3 questions, but just a follow-up on Eduardo's comment. And I think it's really important what he just mentioned about the non-reproductive context versus the reproductive context. We have shown in zebra finches, indeed, that the social relationships that are formed in the non-reproductive context, so without any nest boxes or reproductive material, they predict the breeding pairs that are going to be formed during the reproductive context. So in that sense, even though, I totally understand the same-sex tests that we run and everything, I'm kind of doing the same with the guinea pigs. Just because in mice, it's very difficult to control for the reproductive season. They can just, they can get just like very easily into reproduction, even though they have these cycles. But so just to mention that, yes, during the non-reproductive season, you can actually be looking for who's going to be your mate later on when things are ready to reproduce. So maybe that's something to look into. And my question, it's related to, and I kind of like also something that Eduardo just mentioned, differences between gerbils and the spiny mice, in terms of their social organization, so how different is that in terms of like, are they harem type, or is it a multi-male, multi-female group for both of the species? And how different is the group size between them? I know we don't have that many data on the, like from the field or behavioral observations on the spiny mouse, but I think that might tell us something about why these group size tests and kin, non-kin are actually different between these two species. And I think, yeah, you're back.
I'm back. Okay. Okay. I believe I.
But no, yeah. And then you're. Yes.
How about now?
Yep.
Yes.
Okay. Sorry. So I believe I caught the last of your question, which was basically how are the group structures and mating systems different between the spiny mice and the gerbils?
Yeah.
So with the gerbils, they are socially monogamous. So males and females will form a pair bond, and then they're going to have in their little burrow 1 to 2 litters of offspring. And then the offspring will disperse at some point. And so it is like everybody is very, very much related, small family groups. And then with the spiny mice, it is more of these just kind of a communal breeding system where it's usually, it's always more females reproducing. So you get just more females to, that's a lie. It's more males to females. Nope, it's the other way around. It's more females to males.
Or females to males. Mm-hmm.
And you're just kind of in these larger groups. We don't have the field data on their dispersal. If they're like other communal breeders, then both males and females can disperse, but it's mostly males that would do the dispersing. In the lab, you know, when it comes to like how flexible are their systems, like in a lab, you know, this is more artificial. It's not in the field. But with gerbils, you can't really put 2 adults of the same sex together. They're going to fight relentlessly. With the spiny mice, you absolutely can. So at Emory, we're a bit restricted on the cage size that we can use right now. I hope that's going to change. But at our colleagues' lab in Kentucky, Ashley Seifert, he has these nice large cages. And it really is just puts 20 to 30 mice, related, unrelated, and just puts them all in there together. And they're quite content as a group. And so they're just much more flexible. And then when it comes to the breeding groups, you just want to have more females to males. So mating systems are very different. You've got communal breeding and social monogamy. And then it's the group size is based on the mating system. It can only get so big in the gerbils where it's going to be larger in the spiny mice. I think for the spiny mice, a lot of it comes down to space as well, that if you can give an animal more space, then they can be part of a group, but they can always escape maybe a bully that exists, or they can choose to be less affiliative during certain times of the day. And then when it comes nighttime, like when they go to sleep, they all find one little igloo that they can all shove themselves into. It's pretty remarkable in our colleagues' lab, because he's got these big, just hollowed, they're pipes, like the big kind of white pipes, and he cut a little hole in them so the mice can go in there, and like you can like lift up this pipe and like 30 mice come spilling out. Can't do that with the gerbils.
I see. Okay. Well, but that's really interesting in terms of like what we'd expect in terms of in their proximate mechanisms, which is kind of cool. Which brings me to my last question that is just related to sample size. So when you want to look at these differences between kin, non-kin in the expression of all these neuropaths, how many mice do you actually need to sort of make a good case?
Yeah, so we usually find that like a bare minimum of 8 per group. Your effect sizes are much better if you can get up to 15 per group. So it's, you know, we can get away with surprisingly small sample sizes. The more specific you get in what you're looking at. So if you're looking at like oxytocin co-localizing with FOS, you can get away with a smaller sample size than you can if it's just FOS, because the FOS could be responding to a number of different things. And so kind of like the more specific you get, you're more likely to pull out effects. But if it's looking more broad at just like FOSS alone, then usually we have the most success with a group around 15 animals per group.
Interesting. Thank you, Aubrey. Eduardo has another question.
Yes, and actually, Adriana, your question has given me an angle to start this. Because when you asked what was the sample size, I was going to answer 8. Those were the 8 dots we had for each panel. And I'll be totally honest, Aubrey, I didn't buy it because, and then we can talk about effect sizes and power and all those things that we're meeting with Aubrey tomorrow. I think we'll talk some more about it, but I really hope I convince you to explore moving away from this dichotomous way of evaluating the evidence. But going into something more exciting is, what do we know about the relationship between any of these neurotransmitters and their effects? The assumption seems to be that it's linear. So in assessing the effect sizes, in looking at the differences in the graphs, in assessing whether there's an increase or non-increase, it seems to me that the assumption, I don't know whether it's backed up by our understanding of really how these neurotransmitters work, is that the increase on their effect follows a line. And that may or may not be true. That's a question for you. But then for the graph that you showed us, I mean, I'm assuming that with just 8 of each, the best you could do was some kind of non-parametric, very, very basic Mann-Whitney or Spearman. And those dots did not seem to me compelling that there wasn't much there. I'll be totally honest. I mean, that's where we get at this point. Already 12:30, we might as well ask the questions we feel like asking, and we're just having a dialogue. So first, do we know the nature of the relationship between, say, functional connectivity or any of these neurotransmitters and their effect? It's been assumed as linear, correct?
Yes, if I'm understanding your correction correctly, that if there's, it depends. So like if you're thinking about oxytocin, there are studies that use like microdialysis that can measure real-time release of oxytocin. And when you see two animals interacting, most of this work has been done in rats. Depending on the context, you'll see, I think probably the most work's actually been done with moms parenting offspring. And if you record oxytocin release in certain hypothalamic regions, you see as a mom goes to interact with her pups, you see this nice big spike in oxytocin. So I think that says, yes, this is a linear relationship. I think that's what you're asking.
What I was trying to understand is it the case that, because it could be that there's a threshold, and really whether you have the spike or not doesn't follow a linear response, but you get to a threshold after which really any increases on release are not going to change the behavioral phenotype that you're evaluating. And that's something that I just don't know, because it's about neuroendocrinology that I don't know a thing. So that was the question.
Yeah, I think it's going to depend. There's got to be a threshold for most things at some point. So whether we're hitting that, we certainly can't tell from these methods.
Thank you. We have some we have some, I don't know if they're students, but we haven't heard from some of the people in the audience, and this is when we definitely want to, right, Adriana? I mean, Sarah.
Right, I was going to say the same. It's like, Jonathan, you haven't really talked. Anne-Marie.
Julian Julian wants to ask a question now.
Hello, Aubrey. Thank you very much for your talk. Very interesting, because I think many of us are not in the neurological base of things, so it's really interesting. You hear me fine? Yeah. Yes. Okay. Perfect. So I have a couple of small questions. So one question I have about the setting where you put the group versus the two animals, and then they have to decide where to go in order to see how pro-social they are, what their choices are. How do these gerbils deal with being packed 10 individuals in one cage?
Yeah, and so What we'll do, we have kind of two different ways of doing this. Sometimes it's a long kind of container that has divided spaces such that there's one animal, they're all clustered together, each in their own little compartment in a larger container. And the gerbils are fine with that. When we put a bunch of gerbils in one container together, we're taking a bunch of siblings. And so it's like all those siblings, they know each other, but they're going to be novel to whoever our subject is. That way the gerbils aren't going to fight in there. If we were to put a bunch of adult novel animals in that tight of a space, then they would start fighting. So we just avoid that.
That's what I was imagining, because then obviously seeing a bunch of animals fighting, like would be a cue for me not to go close,
Yes, yes, absolutely. Yeah, and so we've done it both ways to try to control for just if you see more animals interacting, that could be there's just more to watch. And so they go over there. One could argue that that is inherently part of being attracted toward a group though. But when we do it both ways of whether all the animals are clustered together in individual compartments or they're kind of freely interacting together in one compartment, we see the same behavior from our subject at least.
Okay.
And you can get that many gerbil siblings because man, their litters are usually like 8 to 9 pups per litter. Massive litters.
Okay, another question is also a bit of a methodological question. Back then, when I was in my grad school, I did work on neurobiology, but how do you know that you're in the right spot when you're entering those fibers, this probes? How do you know that you're in the right spot? Because they're tiny. I remember them being so small.
Absolutely. So unfortunately, we never know until the end of the experiment. So there's with something like fiber photometry, you're injecting a virus that is going to fluoresce so that you can get your signal, but it's also, you can have it fluoresce such that then once the experiment is over, you can look at your brain tissue to see where did my virus hit? And then you can also track the actual track of the fiber that went down. And so you'll be able to see, first of all, is your virus glowing in the right spot? If it's not, then that animal's data is useless to you, unless you now have a control region. That if you get enough animals that are amiss, then a lot of people will use that as kind of a control of, we're looking at the same behavior or whatever an animal is exposed to, and you get a response in your region of interest, maybe the nucleus accumbens, or maybe it's right next door at a neighboring brain region, and maybe you see a difference, which is potentially more compelling.
Okay, yeah, that makes sense. But yeah, that's obviously, how often does that happen? Because I mean, there's animals being sacrificed for that. So is that happening a lot, or do you know by, okay, I enter it like 1 centimeter in that angle, or you're getting it pretty close hit actually?
Yeah, and so we're usually pretty good at it. There's a lot of practice before you start working with real animals. We do have everything's done on a stereotax, so it's very kind of specific of you're referencing Bregma, and so you get really tight, close coordinates. And depending on the surgeon, the hit rate can be as high as 100% that every animal that you target. Brandon is almost at 100% hit rate for targeting the lateral septum and actually the nucleus accumbens. And so you kind of hone in on these coordinates and you get really good, and you get to be that consistent, being able to use a stereotax. There is a spiny mouse brain atlas. It's okay. It sort of maps onto the coordinates that we've obtained. There's also a gerbil brain atlas, which is actually pretty great. And so we've done enough with both gerbil and spiny mouse brains that Brandon in particular, a student in my lab, is very good at being able to look at the gerbil atlas and go like, this is what it's going to be for the spiny mice. And we can even take brains of animals that are already kind of mounted on slides and look under the scope to be able to get measurements of maybe depth. And so you can get a lot of information before you go in, so that you're not just going in blindly and needlessly wasting animals.
Okay. And I have many like tiny questions, but one other question would be how, going along those lines about personality traits again a little bit. I think people are fascinated by that. How much value you think would be of getting those, when you showed us the social networks that you created, if you take those individuals that are peripheral and do not interact, like how much value do you think they would have to inform about an ancestral state, or do you think that this is already a secondary development, that there are some that are just not very social?
Yeah, I don't think we'd be able to determine anything about an ancestral state. But for any species, it's good to have a lot of variation in behavior and different strategies for how you navigate a group. Let's say in a really aggressive intruder somehow does come into the group, and the animal that's hanging out in the periphery is probably going to be more likely to survive. And so it's just good to have a variety of strategies for interacting in groups. And so I think it's very natural to see animals ranging from I'm in the group, but I'm a little bit on my own, all the way to like I'm in the group and I know everybody and everything in between. So that I think that's just natural variation that you're going to see in all types of groups.
But in terms of their neural responses, I'm thinking more in that realm. Like, would that be like, okay, this is what a solitary species would look like, likely?
I don't know, given that they've still evolved to be in this large group living species, I think it'd be a stretch to assume that's maybe what a solitary species would look like. I think there could potentially be a big difference from like the neuroanatomical profile of a solitary species versus just basically an introverted mouse that's in a group living species. But we can't know for certain.
Okay, thank you very much. Thank you.
Thank you, Julian. Great questions. And we have Anne-Marie.
Hey, and I'm Anna-Marie I'm a postdoc at the University Católica in Santiago, Chile. I had a question about like for your sample size. I'm actually like surprised with like how small your sample size is. Is that like due to like the price of like putting these tags in, or like I have no idea. So yeah, can you kind of like talk about that?
Yeah, this does come down to doing neuroscience, is that you have to collect the brain from the animal at the end of the day. And just none of us can justify taking the brains of the animals that would make for the ideal sample size. And so it's every neuroscientist is working within limitations of being able to hopefully find some answers and to obtain significance. Maybe there's a decent effect size, while balancing that with the ethics of you are using animal life for this. So everything is kind of walking that balance, that tightrope. But our sample sizes are pretty standard for behavioral neuroscience like this.
Yeah, so but you also talked about like that you were able to like plug something in, like and then you still have to like euthanize the animals afterwards.
If we're doing like an implant, because we need to verify that we actually hit the right region and are recording from the region we think we are, then unfortunately, yes.
Okay. Yeah. Thank you so much.
Thank you, Anne-Marie. Eduardo, I'll skip you and I'll let Nancy to ask her question.
So I didn't quite catch that, Adriana. Is this my turn?
Your turn, yes.
Okay, good, because it's a follow-up of what Anne-Marie just asked. So really cool stuff, Aubrey. I didn't know you had switched systems.
Yes.
I didn't know you had worked on so many systems, including the finches either. But my question is about sample size. You said you had groups of like 8. How many groups? Or is it one group and you look at every individual in the group?
I'm going to even go back. So our standard sample size for a lot of these things is 15 animals per treatment group. For like the group size preference stuff with the sexes combined, I think we're actually at 20 animals per group. For the kinship ones, it's for the kin study, it is 8 animals per group. But for the other ones, we're usually at 15 or higher.
Okay, so do you just look at every animal within one group? Is that what you mean when you talk about your sample size?
So like if it's an immediate early gene study and like one group of animals that has 8 animals in it, each one of them is exposed to kin, and then our other group, each of the 8 animals in there is exposed to non-kin, and then we're just comparing exposure to kin and non-kin, so it's 8 per treatment group. That makes sense?
Yeah, now I understand what you're saying, because it seems from some of the stuff that we've done with the statisticians here, now I know you asked 2 statisticians, you get 2 somewhat different answers, sometimes totally different answers, that there's interdependence because they're within the same group, because they had advised us with the voles that we needed multiple groups. Now, and so when we report sample size, it's like number of groups or number of families, and usually 1 or maybe 2 animals within each family or group.
For at least the kinship, the IAG study, there is no dependence between the subjects. It's everybody's their own little subject. Everybody has their own unique exposure.
But they're all, oh, okay. So they're
if you're referring to like the social network analyses, then where we're looking at the group behavior, is that what you're talking about?
No, I thought in the experiments they're exposed as a group, but maybe I misunderstood that part.
Sorry, that must have just been the way that we use this word. No, they're not as a group exposed. It's in the immediate early gene studies, it's always a dyad. And so it's, your one subject is exposed and interacts with kin. Their IAG study is done. The next subject number 2 interacts with somebody. And so every subject has their own interaction. They each have their own brain data. And so there are 8 animals with their independent unique data per like treatment group is the kin or a non-kin.
Okay. That was my misunderstanding then. Thank you.
Thank you, Nancy. Eduardo?
Thank you, Adriana, but if you need to go, please, I mean, we can either go, make me host, and we keep chatting if you want, but you want to close the online and be done with it. I know how it is. You have something coming up at 1. So it's your call. You're the boss here.
Should we stop the live stream and then we can just stay chatting? Good. Okay. So Aubrey, thank you very much for your talk. It was amazing and for a great discussion. And we hope to see you around at some point in one of our seminars. And so as for now, I'm going to close.